Thanks, David, for the invitation to speak today. I'm really happy to said. I guess one of the positives of all this business is that we have the opportunity to connect online, and I think that's been one of the few positives of the situation. I also didn't realize how bad my photo looked there, so I must change that. So today I'm going to talk about two studies, and the studies are related to, the topic I've chosen to talk about today is the role of testosterone in muscle hypertrophy regulation in young and old humans. And I kind of apologize in advance at the moment. These studies that we've done do focus on male subjects, but I'll be happy to field questions around the potential role of or the implications of these data in terms of females. Sorry, let me move on. So to give a bit of background, what I'm going to do is talk a little bit, first of all, about the role of potential role of exogenous testosterone. And what I mean by that is the provision of testosterone therapy to an older age group. And in the second half of the talk, I'm going to speak more about the role of endogenous testosterone as it pertains to muscle hypertrophy regulation in younger subjects. So some background information, first of all. In men and women to an extent, testosterone levels decline with age, obviously. This is due to various different aspects regarding the gonadal pituitary sort of axis, whereby testosterone levels decrease through reduced activity of the Leydig cells of the testes in men. And at the same time, the binding partner of testosterone, SHBG or sex hormone binding globulin, that also increases. So you get both a reciprocal decrease in testosterone bioavailability through reduced impaired secretion through the endocrine changes with age, but also at the same time you get an increase in the quelling signal from SHBG, reducing overall free testosterone to bind to its receptors. It's been associated with many things. Of course, we're talking about muscle today. So declined muscle strength, declines in muscle mass, of course, decreases also in insulin sensitivity, because of course muscle tissue is one of the primary tissues that is dealing with postprandial glucose deposition, but also an increase in fat mass. And so it has many, of course, pleiotropic effects in terms of human physiology. And if we kind of look, this is what the study I'm going to present is clearly not the first study that is administered testosterone or analogs to older individuals. There have been studies suggesting that if you provide testosterone, so if you look at graph B, bottom left, at different doses, there is a sort of dose-dependent response in some of the physiological parameters, which include things like strength and body composition. These studies vary hugely in terms of things like duration, obviously, and dosing. And I know questions will arise around the potential for things like side effects with things like testosterone. I'm, again, happy to answer any of those sort of questions. One of the reasons as well, apart from the physiology of aging in terms of the declines in testosterone that you see depicted in A, is that also many studies, including larger scale composite meta-analyses, have shown that there is a decline in the ability for aged muscle to grow, and strength improvements, can be, in terms of relative improvements, can be fairly well maintained, but there is certainly a decline in the ability to hypertrophy older muscle in accordance to many studies. So one of the things that we were keen to sort of look at was the potential for short-term, in particular, testosterone therapy on the backdrop of exercise in relation to the fact that older people seem to elicit this reduced anabolic responsiveness. So just before I get into the data, just to revisit a little bit about some of the basic endocrinology, and these are little snapshots of data that we've generated from papers with much more information, but nonetheless, it hopefully provides a sort of basic framework as to where I think we're at with this. So here's some data on our groups of younger and older adults. And by that, I mean typically, sorry, because it could be in different places you might call younger one age versus another, and older the same thing. I'm typically talking here around about 20 to 30 as in younger, as it were, and 70 plus in older, at least in our lab. And most of these, by the way, are free of obvious morbidities in terms of at least clinical manifestation. You can see there's some classic characteristics in endocrinology. In particular, you can see if you look at the top left and top right, serum IGF-1 and testosterone are significantly lower, as you would expect in our older age participants. Myostatin is not. Myostatin, if anything, actually is higher in younger than older subjects, which, if you think of its role as a growth antagonist, is kind of contrary to some of the earlier data. But we routinely find that there is no increase in myostatin in older age. Just note the red loop around the top right graph, because the other thing we've looked at is how testosterone concentrations change with acute exercise. And again, it's well established that IGF-1 growth hormone, for example, testosterone, will increase with muscle activity. And of course, that's, as I'll get to in the second part of the talk, that's thought to be a process that's important in potentially the regulation of muscle hypertrophy. In this particular study, bottom right graph here, you can see that there is albeit I know that I fully accept the subject numbers are relatively small, but nonetheless, there was a significant association between the acute change in plasma testosterone and the hypertrophic response thereafter. So we were keen to look at the effects in this first study of supplemental or exogenous testosterone provision in older age alongside resistance exercise or in the presence of a placebo, to determine whether or not it might impact some of these endocrinological features of aging and poorer hypertrophic response. So that's our study design. It's 6 weeks. You can see from the top left there that we provided either placebo injection or testosterone, so a saline injection or testosterone in the form of this Testosterone, which is a clinically approved drug here. We did whole body resistance training. So what I mean by that is loads of different exercises, lower and upper body stuff, 3 times weekly, 80% 1RM. We gave a bolus and then top-ups of deuterium oxide, so we could estimate chronic protein turnover. Again, happy to answer questions. Maybe, hopefully, some of you will be familiar, if not all, will be familiar with these approaches. We did DEXA scans for body composition, ultrasound for local or lower limb, in particular, vastus lateralis muscle thickness. We biopsied, of course. The idea was that we could capture potentially some of the molecular changes that were going on. But we also did oral glucose tolerance tests before and after, because reflecting the fact that testosterone has been implicated in insulin sensitivity as well, which of course does decline in older age. So let's go on to some results. So first of all, we wanted, of course, to see that when we provided the testosterone, that it was having the desired impact. These were, I should have mentioned on the previous slide, these were non-hypogonadal men. So in other words, they didn't achieve they weren't clinically hypogonadal. Nonetheless, of course, as I showed you in previous slides, they had lower testosterone levels than their younger counterparts. When we provided the Testosterone by injection, regular injections, you can see the effect there. And actually this quantity of testosterone, which actually, if you delve in the literature, it's quite messy because serum versus plasma concentrations can be quite different. So it's something to sort of think about. And when we were trying to determine clinically where we were in terms of testosterone, it turned out we probably were in the sort of, youthful physiological range rather than the super physiological range of testosterone. I thought that's kind of important to mention. So we're not in some, hyper testosterone state here. Recently in the literature, and not just recently, but for a number of years now, there's been an implication of the androgen receptor in muscle being acutely regulated by exercise and also being potentially linked to adaptive responses. So of course, we of course looked at this. So this is actually an mRNA measure obviously by PCR. What you can see there is that in the testosterone group, you saw a chronic upregulation of the AR, whereas in the placebo group, the older placebo group, we did not see that. So this was suggesting that the testosterone was indeed having an effect in sort of self-regulating the androgen receptor expression within muscle. And as I said, there is an interest around that general area of how the AR might lead to adaptation. So we then, if we then look at some of the body comp stuff, so there's quite a lot of graphs here, so I'll take my time a little bit. This is DEXA. If you look at A and B, fat-free mass, which obviously, again, is not obviously a perfect measure of lean mass or muscle tissue. But you can see there that in the older group, which is what we commonly find, especially over a shorter period, again, this is, there's a lot of literature out there about adaptive responses in age, and some suggesting there is more adaptive potential than others, confounded somewhat by intervention periods, training loads, etc., etc. Nonetheless, over the periods that we measured, and one of the reasons we measure over such a short period is we feel that, if we're going to intervene in a rehabilitative means or prehabilitative means, then we need a relatively short window to have an effect. We find over 6 weeks that in the placebo group, which is what we often see in our older group, we don't see any notable change. Again, we accept fully the small group sizes. Nonetheless, there are changes are fairly small, if any. Whereas in the testosterone group, we see a significant increase in fat-free mass. When we look at total fat mass, again, testosterone is associated with lower body fat oftentimes, and you can see there that there is a reduction in total fat-free mass, which again would be somewhat associated with an increase in muscle mass due to, of course, things like its impact on metabolic rate, basal metabolic rate at least, and other things. So again, that change is only seen in the testosterone group. We also had the ultrasound measurement. You can see that the only significant statistically we saw was in the testosterone group and less so in the placebo group. The graph D represents a sum strength. What we did here was quite simply to distill it down, because of course we measured lots of different strength parameters. We looked at the sort of percent change across different exercises, and we see that the testosterone group increased more than did the placebo group. And when we look at the relationship between the sum strength and the fat-free mass change, we also see in the testosterone group there is somewhat of a relationship, again, except in the low numbers. So we went to move on to have a look. On the right-hand side, you can see here that there are a bunch of stuff that relate to those adaptive responses that are well established: translational capacity, efficiency, leading to, of course, protein accretion and eventual muscle hypertrophy. So we'll have a look at some of those data now. So using the D2O approach, again, just to simplify in the interest of time, week 1 to 3, week 1 to 6, I wouldn't worry too much about, in terms of the fact that by taking biopsies, of course, sequentially at different times during the intervention trial, we're able, of course, to capture, windows, as it were, of synthesis rates using this approach. But really just focus on this 1 to 6 week, which is effectively the full period of the intervention. And you can see there that in the, as we've shown before in older individuals, over 6 weeks, we don't see a great deal of increased incorporation, whereas in the testosterone group, we do. We're also able to calculate a bunch of other things through having the FSRs through that long-term D2O approach, but also DEXAs. So we can calculate things like absolute synthesis rates, fractional breakdown rates, and fractional growth rates. And again, just please refer to the paper if you're interested in those methods on those approaches. But suffice it to say, to summarize in the interest of time, what we actually see is that as well as synthesis going up, actually breakdown appears to go up too with testosterone. So what you end up with is a heightened protein turnover, but whereby synthesis still exceeds breakdown. So it seems to accelerate protein turnover, which anyone that's interested in proteostasis of aging would probably recognize that, you know, impaired proteostasis or autophagy related processes are associated with, you know, muscle dysfunction, but also more generic aging features in physiology. We also had the opportunity to look at how acute signaling pathways that, again, are very well established, respond during acute exercise. So one thing I didn't really give full information on when I presented the protocol was that we also were able to, at certain time points, and here you can see baseline week 1, week 6, for example. So baseline was, you know, obviously basal, needless to say. Week 1 was after a week of the testosterone therapy, and week 6 after 6 weeks of the testosterone therapy or placebo. What you can see, hopefully, from these graphs is that we took a biopsy about 60 to 90 minutes post each exercise bout, and we looked at whether the provision of the testosterone was able to enhance the signaling response. What you can actually see is that it was indeed the case that happened. So again, it's not, there has been studies showing there is attenuated signaling responses in poorly hypertrophic muscle, including in aging. And here, hopefully, you can see that, say, signals like AKT, RPS6, which is a substrate of P70, SXK1, downstream of TOR, were reduced in comparison to the placebo group. Increased rather. Sorry, in comparison to placebo. I'm moving to the next slides already. When we looked at things like what we call translational capacity, which is the ribosomal, so, you know, the amount of ribosomes, or if you will, which of course, whatever 80+% of our total RNA is ribosomal. So we tend to assume that total RNA concentrations tell us something about ribosomal capacity. You can see that this was only significantly increased in the testosterone therapy group. We did some gene stuff too. We went and looked at a bunch of things like basic helix, loop helix factors, transcription factors involved in obviously muscle development, but also postnatal muscle development and regeneration. And also things like IGF-1EA, which is the muscle splice form, or one of the muscle splice forms, I should say, of insulin-like growth factor. And once again, we find that it's increased more in the T group than the placebo group. We also, as I said at the start, looked at the oral glucose tolerance testing profile. So from this, you obviously give a load of glucose, then you take regular blood samples and you measure at least glucose and insulin. You can then map things like, you know, time-dependent differences in insulin secretion phases, let's say, and glucose clearance. And what you can see if you look at the graph A here is that it looks no different in terms of glycemic control in response to that load. If you look at graph B, what you do see actually is that the testosterone group tend to, as you can see, hopefully have a lower overall net response in terms of insulin than the placebo group, and that is indeed significant. So this is suggesting that to maintain that glycemic control, less insulin secretion may be required. If you look at C-peptide, which of course is produced upon insulin, you know, insulin production, you can see that in the testosterone group in C, it's lower than in the placebo group. Again, suggestive of the notion that lesser insulin secretion is required to maintain that tight glycemic control following the glucose load. Using equations to look at proxies of insulin sensitivity, you can see the C-home index on the right and D. This is taking basically certain samples from early on in the OGTT phase, whereby you can calculate insulin sensitivity proxies. You can see here that also in the testosterone group it's improved, but it's not so in the placebo group. The OGT 1, 2, 3, I should have mentioned here, I should have been more clear. We took the OGTT 1 is baseline, 2 is after 1 week of just provision of, and 3 is after 6 weeks. So you can see that really quickly it seems to have an effect upon these parameters. So there's a graphical conclusion here. Again, I'm looking at my clock and going, oh, heck, I've been speaking that long already. So I better move on to the second phase. Please, this is the summary of those data. But again, this is just effectively what I've just described. So I'll move on. So let's move on to youth in terms of testosterone. So again, establish that testosterone increases associated with increasing fat mass, increasing muscle strength, fat-free mass, sorry, increasing muscle strength and decreasing fat mass. And I've shown you data already that suggests that, you know, testosterone exogenously alongside resistance training, and this is obviously in the field of not just aging, but also doping, is having some positive effect in terms of muscle mass. Question is, what about endogenous testosterone? So this is a study from a colleague, collaborator, Stu Phillips, obviously a fellow Canadian for those out there, fab researcher, obviously. And this is just one excerpt of data whereby, you know, using a really cool model, they were able to show that if you exercise a big muscle mass alongside or against a low muscle mass, and then take, so say for example, you exercise the legs and you exercise the legs and the arms, but you then take biopsies only from the arms. If the systemic response is greater when you have that greater activation of muscle during the arms and leg exercises, therefore, if that systemic influence of testosterone were so important, one would expect maybe the biceps to grow more. Neatly in this study, they showed that was not the case. So in the high hormone and low hormone environments, there was no difference between strength gains or let's say elbow flexor cross-sectional area. Many other studies, but this cast doubt on the role of endogenous testosterone. So we thought that we'd do a mechanistic human trial. So this is what we did. Again, I'm going to keep this really brief because otherwise I'll get told off. But basically, it looks a bit similar to last time, except this is younger subjects. Again, relatively small N, around 8 per group. We provided this time not testosterone. We provided a drug called Zoladex, which is a gonadotropin-releasing hormone agonist. So basically what this drug does, I'll show you in the next slide, is it creates asynchrony between the Leydig cells and the hypothalamus and pituitary systems. And therefore, what happens is eventually you actually get a marked suppression in testosterone. And this is used, for example, in things like prostate cancers to try and limit any risks of testosterone propagating tumors. Protocol looks much the same otherwise. So I'm going to skip forward. 6 weeks again, whole body exercise, placebo or the drug, which is going to drop testosterone. So here's the data. Here's some data. Left-hand side shows you what I've just pretty much described. Middle graph shows the testosterone concentrations. The gray area is into the sort of, you know, toward the hypogonadal end. So we're basically pushing people toward, you know, hypogonadism rather than zero testosterone. So, you know, they're effectively becoming hypogonadal. You'll see the little spike after a few days. That's because it's an agonist, but eventually it works by antagonizing through that, creating that asynchrony between the Leydig cells and the hypothalamus. Again, we looked at androgen receptor and we see that in the placebo group, in the younger men, it goes up, and in the Zoladex group, it does not. Body composition, fat-free mass increased in the placebo group as expected, as did muscle thickness. If you look at graph C, as body fat decreased, if you look at graph B. But at the same time, we actually saw that provision of the Zoladex, which of course pushed them into the hypogonadal state, completely blocked all of the adaptations that we were able to see in the placebo group. Once again, looking at some strength, you can see the percent change in the placebo versus Zoladex. So it's not to say they didn't get any change despite the lack of hypertrophy, which again is fairly consistent with the notion that you can, of course, get neuromuscular adaptations in the absence of favoring strength in the absence of hypertrophy. Nonetheless, you can see here that it's suppressed in the Zoladex group. And similarly, as I showed before, if you look at total testosterone levels and some strength changes, of course, in the group where there was variable testosterone in the placebo group, or natural variation in testosterone, there is a somewhat of, again, even though it's a small study, a correlation between some strength change and total testosterone. So then on the right-hand side, we went to look at this stuff again. You can see here that using the D2O approach, again, as you would expect, over the 6 weeks of training, we can see that protein synthesis day to day, this is a percent per day, as you can see on the y-axis, increases in the placebo, but not in the Zoladex group, in keeping with the data that we had, of course, in terms of mass gains or lack of.
That's about 1 minute. Okay.
No worries, Tara. I'll be done. Thanks. Well, maybe 2. Breakdown was also seemingly suppressed in the Zoladex group. And overall, if you look at the overall net fractional growth rate, and again, I would just refer anyone to look at the paper from the first half of the talk, you'll be able to sort of see the derivations of these calculations. But again, protein synthesis is suppressed, and therefore muscle does not grow. Looking at some signaling again, you can see on here that, again, this is the acute bout effect, that the induction of hypogonadism actually blunts the acute induction of AKT mTOR signaling. Again, this is just a small snapshot from a larger data set. Similarly, if we look at RNA concentration gains, accretion, IGF-1 EA again, but EC is also present here, which is the so-called mechanical IGF-1 thing. And a couple of the basic helix loop helix factors, you can see again that these tend to be suppressed versus in Zoladex versus placebo, apparently explaining the response. Finally, before I move to the conclusion slide, the glucose insulin data and from the OGTTs in our younger individuals. Once again, you can see that the glucose excursions don't look much different across the groups, but there was a tendency toward the Zoladex group blunting the apparent reduction required in insulin signaling to maintain glycemic control. And this was also fairly clearly seen in the Cedar Home Index, whereby only did the individuals who were taking the placebo, not the Zoladex, show some improvement. So that's the graphical abstract. Again, Tara's already shouted at me, so I shall stop there. Hopefully that's all reasonably clear. There's my summary thoughts. T provision to older men enhances hypertrophy, maybe represent a useful short-term adjuvant therapy, for example, during rehab or prehab, before hospitalization for elective surgery or the like. T depletion in younger men attenuates muscle growth, and that suggests that at least endogenous T sufficiency is an indispensable factor for muscle hypertrophy that cannot be rescued by alternative mechanosensitive pathways. And I don't think that conflicts at all with someone like Stu's beautiful study at all. I just think that if you're in the realms of hypogonadism, that can be a real problem in terms of muscle growth. Thank you, and I'm sorry for talking a little long.
Hey, thank you, Phil. If you have any questions, please enter them into the Q&A. We have one question from James McKendry. Phil, great talk, cool data. With testosterone administration, breakdown is increased, but with testosterone suppression, breakdown is suppressed. How do you reconcile these perhaps unexpected findings?
Yeah, so I think that it was really surprising when we saw that when we provided the testosterone to the older folk, that breakdown increased. But what I must say, James, and thanks for it's a really cool question as well, is that these are basically estimates based on calculations based around, DEXA accrual against the D2O data. So they have to be taken with a huge pinch of salt. And again, we encountered this on review such that people said, well, actually, loads of cell models or animal models have said testosterone reduces breakdown, which is, as you say, what we do see. But in another instance, So I think that, you know, I think I don't have the full answer to that, but we also have to take those, you know, those extrapolations with a pinch of salt.
Great. Okay. Thanks, Phil, very much for your talk.